organizers for this invitation. So let me see how this works. Okay. So I came here to share some results that we obtained at Vanderbilt University in collaboration with Dr. Gabor. And we are looking at some anti-aging effects of DDW in Caenorhabditis elegans. So just a brief outline of my talk. I'm going to talk a little bit, introduce you to C. elegans as a model for aging. And I'm going to talk about a little bit about manganese toxicity. It was the model that we used to induce aging in this animal model, and going briefly to results and discussion. So just an introduction, what is Caenorhabditis elegans, or C. elegans, as I'm going to refer during this talk. It's a very small nematode, so it reaches 1 millimeter long as an adult. It has a transparent body, and it feeds with bacteria, and it can be grown in liquid media or in agar. It's a solid media. It has a large brood size, so approximately 300 progeny per hermaphrodite. So this is very good for high throughput screenings of toxicants and also antioxidants or protective compounds. It has a very short lifespan of approximately 18 days in the wild type. And it also has a very short reproductive cycle, which is represented here. So the reproductive cycle, it's approximately 3.5 days only. So as you can appreciate here, the embryonic development goes through 14 hours, and then the egg hatches, releasing L1 larva, which goes to L2, L3, and L4 stages in very short period of time. And during this period, the worm matures, and especially the reproductive system matures, and then reach the young adult that is able to reproduce, restarting the whole cycle again. So something that is very interesting in this pathway, in the cycle, is that when this first larval stage, when the animals are in a starved plate, so there is no bacteria to feed with, they go to this dower larval stage, which these worms can survive for approximately 3 months, which is more than the triple than the regular lifespan. So this is a very interesting pathway. So many people are working, so many researchers are working with this dower larval stage because it can be a key role for aging and longevity. So something else that's very interesting is that 60 to 80% of the worm genome, it has homology with the human genome. This is very interesting for these studies. And also something very interesting is the ease of genetic manipulability that these worms provide. So it's very easy to knock out genes and observe the effects of this lack of genes, and also to generate transgenic lines. For example, you can insert a green fluorescent protein to a promoter of a gene, or to a protein by itself, and you can see the pattern expression of this protein using an immunofluorescence microscope, as you can observe here. So disease of genetic manipulability led to the findings of several pathways, and one of the main studied pathways in C. elegans is aging. So C. elegans is an ideal model to elucidate causes of and molecular mechanisms of aging, especially because of that short lifespan, and also the genetic manipulability, of course. And what is very interesting is that as humans, C. elegans experience age-related neurodegeneration, sarcopenia, which is the loss of muscles, and also lipofuscin accumulation. So as you can appreciate here, we have a young adult, so you can see the body bend, so this worm moves freely and looking for food search, new food sources, And as this animal ages, as you can observe here, it changes completely the morphology. This animal is still alive, it's still feeding, but it can't move anymore to search for new food sources. And as you can observe here, this is the same animal. It's an immunofluorescence figure, and you can see that there is accumulation of lipofuxin, which is very similar to what it happens in humans. You can see accumulation of lipofuxin as they age. So there are more than 50 genes that were found to be related to this aging process. And the one that's well studied in worms, it's the DAF16 gene. So DAF16 gene encodes a transcriptional factor called DAF16. And this transcriptional factor, it's very important for anti-aging or longevity effects in C. elegans. So under normal conditions, DAF16 is bound to the nucleus and activates the transcription of antioxidants. So DAF16 belongs to this cascade of the DAF2 insulin-like pathway, and this cascade controls dauer formation, progeny production, stress response, and also lifespan. So if you understand, DAF16 increasing the transcription of this antioxidant proteins, it would increase lifespan. On the other hand, when insulin-like ligands bind to the DAF2 receptor, it triggers a phosphorylation cascade, which includes kinases like AGE and AKT, and as a consequence, they phosphorylate DAF16, which becomes inactive, and it doesn't go to the nucleus to activate the production of antioxidants. So as you can see here, this figure, it has a lot of genes here, but I just want to show you the square would be the wild type, and when you knock out the DAF16 gene, you have a significant decrease in lifespan. On the other hand, when you knock out DAF2 receptor, these worms with this DAF2 knockout, they live twice longer than the wild types. So this pathway, it's really interesting. And something that I would like to mention is that DAF16 is the homologue of the mammalian FOXO. And FOXO has been shown to be related to tumor suppression in tumoral cell lines. So when there is increase in FOXO, there is tumor suppression. So I think this is very interesting to mention. In our model, we use manganese as an inducer of toxicity and decreasing lifespan. So manganese is an essential metal, but exposure to high concentrations of manganese leads to several cell effects. So it's mainly mitochondrial dysfunction because manganese accumulates in mitochondria. There is energy depletion, oxidative stress, activation of necrotic and apoptotic pathways, and this mainly happens in dopaminergic neurons, causing dopaminergic degeneration. So it's very interesting to mention that manganese toxicities called manganism resemble Parkinson's disease symptoms. So people, they have tremors. So it has been used as a model for Parkinsonism. In C. elegans, manganese recapitulates the effects of mammalian dopaminergic system. It shows as a general toxicity effects, osmoregulation defects, so the worm shows cellular alterations, as you can observe here. There is decrease in the body size, so the worms treated with 35 millimolar of manganese, they show smaller size than untreated worms, and they are arrested in the larval stage, which means that they don't develop as well as untreated worms. And manganese causes decrease in longevity, and that's why we use this model. So we hypothesized that given all the beneficial properties of the depleted deuterium water that we hear so far, especially in anti-cancer research, we hypothesized that DDW would have anti-aging activity in C. elegans. So going to some just to explain for you the treatment paradigm, because worms are so different than mammalian systems. So we use synchronous L1 C. elegans. So the very first larval stage, so they are more sensitive to change, oxidative changes, and etc. And they were treated for 30 minutes to manganese chloride, 35 millimolar. And right after, manganese was washed off, and these animals were treated with depleted deuterium water for 48 hours with 2 different doses. So we use 120 ppm and 90 ppm. So some results that we obtained so far. First of all, we did just a regular lifespan using DDW without manganese treatment, and we didn't observe any increase in longevity when we treat these worms only with DDW. So as you can appreciate here, these are the concentrations that we use. So just the regular water, 150 ppm, then 120 and 90 ppm. So we didn't see any difference. However, when we pre-exposed these worms to manganese and then treated them for 48 hours with DDW, we observed, as you can see here, so controls are these black squares and the triangles are manganese. You can see this shift to the left indicating decreasing lifespan. And you can observe that the two concentrations that we use of the DDW, they were able to revert this decreasing longevity caused by manganese. So this was a very interesting result that we observed, and we decided to go deep to study the molecular, what would be the molecular pathway, what would be the mechanism. So we decided to look at DAF16 pathway once it's widely studied in C. elegans. So something interesting that we find was that manganese causes decrease in DAF16 levels, so that corroborates with the previous finding that we saw decreasing lifespan. And DDW, just when we treated the worms with DDW, we didn't observe any alteration in DAF16, which maybe explains why by itself it doesn't increase longevity. However, when these animals, they were pre-exposed to manganese and treated for 48 hours with DDW, we observed increase in DAF16 levels. So DDW was able to revert this decrease in DAF16 levels caused by manganese. So this was a very interesting finding that we observed. So looking downstream the pathway, we look at manganese SO superoxide dismutase. So this enzyme is downstream DAF16, is one of the proteins that it has its how can I say, the expression increased by DAF16 levels. So again, we observed this decrease in manganese SOD, which is related to the decrease in DAF16 levels caused by manganese. And we observed that at 90 ppm, DDW was able to restore manganese SOD levels that were depleted by manganese. Looking upstream the pathway, so upstream DAF16, we look at AKT, is the kinase that phosphorylates DAF16, inhibiting its activity. And we observed that manganese also increased the AKT levels, which would cause increasing phosphorylation of DAF16. And again, at 90 ppm, DDW was able to decrease this AKT level significantly. So everything connected to the DAF16 pathway. So considering all these results that we obtained, we decided to go deep to look at use genetical approach in order to see if only DAF16 was related to this pathway, to these effects of DDW. And we decided to use DAF16 mutants. So these worms, they don't have DAF16, they don't express DAF16. So by themselves, they are already more sensitive, and they have shorter lifespan when compared to wild types. And as you can observe here, still manganese causes decreasing lifespan, and this is possible because manganese acts in different pathways, not only DAF16. So that's why we still see some decreasing lifespan. But what it was surprisingly was that DDW at the 2 tested concentration was able to revert, again, this effect caused by manganese. So this result indicates that it's not only DAF16 that's involved in the anti-aging effect of DDW. So maybe other pathways are involved and not only DAF16. So this, I think, it's in agreement with several findings showing that DDW doesn't act only in one, doesn't have only one mechanism, but like several in the whole cell. So this last figure is just like to show this decrease in lifespan. So here we just have control wild type, and here this dark squares we have just the DAF16 mutant, and you can see how different it is, the lifespan. You can see the number of days that's highly decreased. But this is just to illustrate what we found. So just some conclusions of this work. So we observed that DDW has anti-aging activity in C. elegans exposed to manganese. This activity relies partially on its ability to modulate DAF16 pathway. So as we observed, it's not only DAF16. The use of mutants showed us. DDW treatment recovered DAF16 and consequently manganese superoxide dismutase levels, indicating that the antioxidant proteins modulated by DAF16 are also recovered by DDW. The manganese-induced increased levels of total AKT were diminished by DDW, suggesting that the whole DAF2 pathway is involved and not only signals downstream to DAF16. So DDW doesn't act only in DAF16, but in the whole pathway. And the use of the DAF16 mutants demonstrated that other pathways, most likely, we believe that SKN1, which in mammalians is called NRF2, which is also related to longevity pathways, may be also involved in this anti-aging activity of deuterium-depleted water. I would like to thank Dr. Michael Ashner. He's my PI, my mentor. I'm a postdoc. I'm here to replace him. He couldn't come, so I came here replacing him. And he and Dr. Gabor, they started this work. I think we started like 9 months, 10 months ago. So it's not a very long time that we have all these results. I would like to thank HYD, Vanderbilt University, and also our NIH for the funding for this work. So thank you.
Thank you. Any questions or comments, please?
Have you ever changed the DDW level during the treatment?
You mean like when I was treating them?
Yes, maybe from 120 to 90 after maybe 8, 9, or 10 days.
So you see that the lifespan is very short.
Yeah.
It's only 18 days. I think if you consider that, if you like consider how long rats live, I mean 2 days is a lot for a worm. Like for 18 days, 2 days, I think it's a lot. We were thinking to try longer, but considering that after that, the worms, they go from L4 to the adult stage, they start to have a progeny. We don't want the progeny to be treated. We want to see the effects on the worm. So after that, they start to have a progeny, and that kind of, it's not good for the treatment, for the lifespan at least. Mm-hmm. Yeah.
Any other questions or comments?
Have you ever tested any other molecules which can compensate the toxicity of manganese? So
Not in C. elegans.
So could you say any example for an other something molecules, compounds, which can have the same effect like the DDW when the C. elegans had been treated with manganese?
Yeah. So like not against manganese, but because we are just studying the toxicity of manganese. We didn't come to see to reverse the effect. That's the first study that we've been doing with DDW. But like in the C. elegans literature, you can see there are several compounds that have been tested to increase lifespan.
Okay, but these you have never tested the
Against
compensation
manganese.
of the manganese?
Mm-mm.
Okay.
Would be a good positive control, maybe.
Okay.
Yeah.
Yep.
Any other questions or comments? If there is no, then we would like to thank all the speakers of morning session for their contributions. And there will be a lunch break, I guess, and we will continue at 1 o'clock. Is it so?